I'm happy to welcome Dr. Sham Diwanay. Professor Diwanay is from Abbasab Garware College and he just retired as Professor of Microbiology. He did his PhD with me when you passed out, 2001 passed out, okay. And his subject was related to immunomodulation, and essentially what he saw was in an animal model where he mimicked chemotherapy in tumor-induced animal. He showed that some of the Ayurvedic plants can actually prevent the side effects like myelosuppression without compromising anti-cancer activity of the chemotherapeutic agents. So he actually induced cancer, he used chemotherapeutic drugs, observed side effects of chemotherapeutic drugs, and after giving Ayurvedic medicine, how they were reversed or protected, especially myelosuppression. This was the whole study. But this study is not the only study which has been done in our group regarding immunomodulation. Because he is going to talk about immunomodulation, I must give you a brief background of this work. We know that there is a class of medicines in Ayurveda called Rasayana, and these Rasayana are mostly class of Ayurvedic drugs, and there are different types of Rasayanas. Or there are Rasayanas which are meant for brain function, enhancing memory, retention of memory. They are called Medya Rasayana. There are Rasayanas which are useful to strengthen your heart. They are Riddha Rasayana. And maybe somebody from Ayurvedic background during your afternoon discussion session should talk about 10 minutes giving broad classification of Rasayana for all of you. In fact, there is one more very interesting work at present going on by one of our colleague. Her name is Dr. Kalpana Joshi, and she is working. We did some proof of concept work here to see, and Gitanjali actually can give some snapshot of what is happening there. But we observed almost 4 or 5 years ago that in a controlled experiments, we hypothesized that typical Rasayana as per the Ayurveda classification will have specific effect on those particular organs. So if there are Ruddha Rasayana are expected to work on heart. Or if there is Medya Rasayana, they are expected to work on nervous system. So this was the assumption. And we hypothesized that if you use these Rasayana in cells, the stem cell differentiation should go in the direction where that Rasayana is expected to act. And in proof of concept study, she observed that that's what happens. And now she is having a major project supported by Department of Science and Technology, wherein she is seeing, observing, studying this differentiation based on the class of Ayurvedic Rasayana. Why I am giving you all this? Because in cancer also, now the focus is on immunity. Globally, the focus of therapeutics actually is shifting from the earlier ways to manage cancer by killing cancer cells alone by using chemotherapeutic drugs. Now the approaches are more regarding how can you modulate different responses, especially immunological responses, so that your body is prepared better to face the situation. So this is the approach actually Ayurvedic therapeutics also follows. And now immunity, immune strengthening, immunomodulator, immunostimulation, or in general immunological interventions have become very important in the field of therapeutics. Now this is also linked with what we normally use term called drug resistance. Drug resistance also has emerged as a major threat globally, and multiple drug resistance. Actually, there is a very good white paper that European Union came out with, which highlighted the intensity of this problem. All this is linked. All this is linked. They are not different pieces. These pieces are linked. And I think today what Dr. Diwanay will talk to you, he will try to connect these links together. So he will talk about immunomodulation, he will talk about drug resistance, mechanisms of drug resistance, and the course which is now you are undergoing, drug transporters. You will be able to then connect all these pieces together over the background of immunology and over the background of mechanisms of drug resistance, and then we can see how this can open new opportunities for research, by which you can use this modulation as one of the tools to counter drug resistance. And drug resistance, I must clarify in the beginning, and he will expand it more. Drug resistance, we are used to understand drug resistance from microbiology point of view, because we are exposed more to that. So we know about bacterial drug resistance, but there is also what Dr. Ambudkar must have talked to you about, cancer drug resistance. So there are quite similarities. So Dr. Diwanay will bring out these issues in this tutorial session, and then we will have discussions. Maybe you can pick up some of those points and bring them back when you are going to discuss with Dr. Ambudkar in the main session. So with this brief introduction, I will hand it over to Dr. Diwanay to conduct this session, broadly designed under the immunomodulation and drug resistance. Dr. Diwanay.
Okay, thank you, sir. I'm very happy to be back here. As sir said, it was, I still remember the date and the atmosphere here, 9th May 2001. In the same hall, I gave my presentation for a PhD. I was very fortunate to have sir as my guide and supervisor from his very first team of PhD students. And that time, the immunomodulation, that word came bit afterwards. So what we wanted to do was check the immunopharmacological responses to certain selected plant preparations, and for that we build up an animal model. Well, I am going to speak something about this. In fact, I have planned my talk in somewhat this manner. Very first, some very basic concepts about the immune system. I am sure everybody is very well aware of all these concepts, all these intricacies of the immune mechanisms. With this background, then I will speak about the work we carried out here. After that, I will go on to immunomodulation, the concept, the definition, possible definition, where it can be applied, and then I will go to microbial aspects, that is pathogenicity. That is the pathogenic mechanisms, and the best tool to study any parasite is bacteria. That was earlier. Now it can be a cancer, particularly if you are using the animal systems. Okay, and then again, after comparing the pathogenicity of bacteria, pathogenic mechanisms related with the drug resistance development, then again we will go back to cancer immunotherapy and modulation of cancer immunotherapy as such. Okay, very basic ones. The definition looks very much legal with and or and so on and so forth. The last part is interesting. Special reference to one that is caused by an infectious agent or its product. So for HS, the microorganisms have been used as a tool to study the immune system, the immunopharmacological responses, and that can be extrapolated to further any pathological condition as such. Why immunity? Because of the standalone features. Of course, self non-self discrimination, which as we will see is going to be bit confusing for immune system as such, because the cancer evolves from the self as such. So, how it is differentiated as non-self, and whether there will be an immune response against it or not. What is advantageous against all other therapies is specificity, because this is going to specifically target the, let us say, the pathogen or the cancer as such, and it is going to leave the normal physiological mechanisms intact. The memory, it remembers what has happened before and accordingly responds whenever there is a recurrence or such. And this, well, may be problematic in some cases, but we will come to that bit afterwards. Well, it's very basic. The innate immune response against the adaptive, it should be adaptive, not adoptive, adaptive immune response. Okay. And gradually the focus from adaptive immune response, which is highly specific, is shifting to the innate immune responses. Again, we will be coming to that. How the immune system works, like a defense system of a nation with barriers as an first line of defense, that is an innate mechanism, non-specific one. Once the barrier is crossed, that can be physical, chemical or biological barrier. That is physical barrier, the epithelial cells, epithelial cell lining all through, the chemical, different secretions and so on, and biological, that is the normal flora organisms, or what we call it now is the microbiota. Once this barrier is crossed, then second line of defense come into picture, which is through cells and soluble factors. The third line, that is adaptive status, that is again cells and soluble factors, and that is classically the specific immune response as such. And there is an hypothetical fourth line, that is question, that what we call is an immunopathological condition or allergic one. The activation of immune response lead to a disease, which is not supposed to happen. This is a bit more of that. Well, the innate immune system, as I said, that is present right from the birth. I can say as an birthright, remains unchanged. Okay. And main response is an physiological inflammation, which later on may turn into an specific one, an immune inflammation, and through a battery of molecules and through non-specifically activated cells, there will be the defense that is achieved. If that is not enough to control the disease condition, then the adaptive immune system measure two types of cells, the B cells and T cells, and I haven't mentioned here, but there are non B, non T cells, or functionally more important ones as an effector mechanism will be the killer cells, maybe simply the K cells or natural killer cells or cytotoxic T cells and so on. All that gives an adaptive immune system which is highly specific. Well, it is quite an complex mechanism. What I want to highlight here is the mediators of this. The chief mediators for innate immune system that are required will be the IL-1, IL-6, products of the macrophages. Depending upon the condition, it can be tumor necrosis factor. Macrophage is a bridge between innate and adaptive immune response. So, if you are building up an immunotherapy as such. The very first what you should look at is activation of the macrophages. Okay. On, you can say, on time scale and the steps in this, innate immunity will be immediate, maybe within few hours. Early induced innate responses to 3 or 4 days as such, and adaptive immune responses will take much more time, okay, consumes lots of energy as such, but the end result has to be removal of the infectious agent. How the immune activation takes place? Okay, I am going to speak about the pathogen, a microorganism, and the most preferred one is the bacterial one. We will leave aside for time being at least the fungal or the protozoan or the viral ones as such. So, it has to come into the system. This has to be remembered all the time, that whenever you want to see the activation of the immune system, if you want to study in the laboratory, if you want to see the expression of the immune system, then it has to be challenged. If there is no challenge, then there won't be any detectable immune response, detectable at experimental level. Okay. So, there has to be the entry into the system, and once it enters, there will be the epithelial surfaces which has many different receptors. The main ones are toll-like receptors. There will be expression of the different antimicrobial peptides, many different soluble factors that are already present. Okay. And the composite innate mechanisms will be an inflammatory response, a very dynamic response. It goes on increasing in magnitude with time, involving more and more systems. Most of these are non-specific: complement activation, kinase system, coagulation system, phagocytosis, and so on. So, the innate mechanisms, as I said, if not sufficient, then you have to go for the adaptive mechanisms, where the antigen will be specifically identified by the macrophages. There are pathways, different pathways, and very broadly you can say there is class 1 pathway and class 2 pathway, or depending upon the location of the antigen with respect to the macrophage, whether it is intracellularly present or whether it is extracellular. Accordingly, the MHC class 1 and or class 2 molecules will be involved in antigen presentation. They will be processed, and there will be either the humoral response, that is in terms of specific antibodies, antigen specific antibodies, and as it goes, it is specific antibodies, which has advantages. When you are constructing an immunotherapy, we want the response to be specific. An antibody, a molecule which requires lot of expenditure of energy as such, has equal advantages as well. The only problem with this is the antigen. We have to recognize the antigen, which is at experimental level somewhat easy when you are using an model system where an infectious organism is being employed to study the immune responses. If it is an tumor, if it is an cancer, then identification of the antigen and finally using that particular antigen is going to be very, very complex, and of course in clinical situation it may not happen at all. Not only that, but like some of the microorganisms, the cancer also goes on changing its behavior with time. So, there can be antigenic modulation, which is, you can say, very characteristic of microorganisms. Like viruses, everybody knows about the influenza virus, the antigenic shift and antigenic drift. Like this, the tumor also goes on changing its antigenic expression. So, many times it happens that humoral responses may not be sufficient may not be satisfactory when you want to construct an immunotherapy. So, you can have the, well, depending upon again lot of factors, the nature of antigen, how it has been processed and presented, there may be an cell mediated immune response. It is cell mediated because the activated cells will be the effector cells, and through the cytokines, different cytokines, there will be T cell activation which can be specific, to some extent B cell activation definitely specific, but activation of the macrophages, platelets, bone marrow, mobilization and all these. The advantage of cell mediated immune response is it recruits whatever defense mechanism the system has in fighting against the disease condition. Okay, so I am sure this was all basic, everybody knows about this. Our work, sir already described in very short. I will take advantage and being here and well speak slightly more about this. We developed an animal tumor model, okay, and we used the sarcoma 180 cells, implanted the tumor in animals. We wanted the animals to be immunologically uniform, so we used an inbred strain of animal, that is Balpsy mouse, the male Balpsy mouse. We characterize the tumor with respect to its growth, and when you characterize it with respect to growth, that all these parameters also help to determine whether it is regressing or not. Progression can be characterized. The same manner the regression can also be characterized. Okay. Then we treated this tumor, and the age old anti-cancer agent, that is cyclophosphamide, still being used. It was used earlier and still being used even nowadays, even at clinical level, cyclophosphamide. We identified a dose of cyclophosphamide, basically an cytotoxic agent. So there was a risk of very severe cytotoxicity that happened in some animal groups, and animals well suffered from this and finally simply became dead because of the cyclophosphamide, may not be because of the tumor as such. So cyclophosphamide dose was identified that significantly brought about anti-tumor effect that regressed the tumor very significantly, and at that dose we observed that there was a very severe myelosuppression, immunosuppression. Everybody knows that the chemotherapy or any other current cancer therapies, it can be an chemotherapy, it can be a radiation therapy, or maybe in some cases surgical therapy, has these side effects. There is very severe immunosuppression. Okay. So now with this tumor model, that is tumor bearing cyclophosphamide treated animals received plant derived preparations, and cytoprotective effect was evaluated using cytological and immunological parameters, and that time we did not have very good immunology lab as such. So whatever can be done in microbiology lab, those systems were used, and I think the parameters, the experimental system used were so effective that when we publish, there are a lot of citations on this, okay, because of the simplicity of these experimental systems. We used 3 plants, but basically focus was on Withania somnifera, ashwagandha, along with that Tinospora cordifolia, and then Asparagus racemosus, okay, Guduchi and Shatavari. So different extracts were prepared, formulations were made. Most of the extract exhibited immunostimulatory activity, but there was also a toxic activity, and these were discontinued from further studies. This was, well, I don't know, but quite disheartening that time, that none of the test material exhibited anti-cancer activity per se, and later on it was found that that is good thing that happened. Okay. So, we could focus more on the immunological aspects. 2 test material exhibited best immunomodulatory activity among different test material. We started calling this as immunomodulatory because you will see that modulation can be on both the sides. It can be immunostimulation. In fact, many times it is considered as immunostimulation, which need not be, because immunostimulation can lead to pathological condition. Okay. So, what we want is the modulation, normalization of the immune system. Okay. Okay, so immunostimulatory activity was found in tumor-free animals receiving or not receiving anti-cancer drugs, that highlighted that definitely these drugs, these plant-derived drugs, were immunostimulatory in normal condition. There was protection from toxic side effects without compromising or interfering with anti-cancer activity to identified test materials, and thus we concluded that these candidate drugs is important in development of supportive adjuvant therapy to cancer therapy to avoid untoward effects. So, that was the work we carried out, and this work was taken further to a great extent by search students, and the immunomodulation with respect to Th1, Th2 responses, balanced Th1, Th2 responses has been carried out. Okay. So, now let us come back to immunomodulation. Encyclopedia Britannica has put a definition. There are lots of definition, but finally this one is more suitable, more appropriate. Okay. So, these are biological response modifier. They modify the response in such a manner that they exert anti-tumor effect by improving host defense mechanism. They do not have direct anti-tumor activity, but they improve the host defense mechanisms against tumor. But Encyclopedia Britannica has also put limitation on this definition, that it speaks only about the tumor. Okay. We will see whether this immunomodulation concept can be extended further to infectious conditions as well. And I should mention here as well that a very good model system for bacterial infectious condition is the tuberculosis, a newly emerging challenge. Tuberculosis, as you know, MTB, Mycobacterium tuberculosis, has developed drug resistance. It is equally wise as that of the host immune system. It goes on changing its pathogenic behavior as to that of the host immune system. So, it is an continuous battle, and that is very interesting. So, whether we can, instead of using directly or using solely the anti-tubercular drug, can we have the immunomodulators that will exert their anti-tubercular effect by improving host defense mechanism? That is an, you can say, hot field at present in immunomodulation. What we tried to define on our part, the immunomodulation, is immunostimulation against immunosuppression by the same preparation. That is, drug induced immunosuppression has to be counteracted by stimulation. In hyperreactivity model, that is, as I said, immunopathological condition, there needs to be immunosuppression. Presently in allopathy, there are two different types of drugs that bring about either the stimulation and suppression, and that effect is chemically defined. Okay. What we look for is whether the effect is carried out by the same preparation. That can be called as true immunomodulation. That is the activity to normalize the pathophysiological processes. And if any such preparation has this effect, then that will be an true immunomodulatory agent. So far, there are no chemical drugs, pure chemical entities that have both these effects. They are not true immunomodulators. There are immunostimulatory agents like levamisole, but it does not bring about the immunosuppression. All cytotoxic drugs are immunosuppressive in nature, but they cannot bring about immunostimulation. What we observed in our study, and that is being continuously explored further, is the preparations from traditional practices, mostly the Indian ones, have this immunomodulatory effect. Okay, coming back to immunomodulation, at adaptive immune responses, it can be either Th1 or it can be Th2, which has to be critically modulated. The Th2 responses that are mostly the characteristics of humoral immunity, where an immunomodulatory cytokine that diverts the response from Th1 to Th2 is IL-4. In other words, should say that IL-4 whenever dominates the cytokine as such, cytokine profile or cytokine cocktail in the system, that suppresses the Th1 responses. On other hand, you have the cell mediated immunity through the Th1 activation, which produces, which liberates interferon gamma, which inhibits the Th2 cell. So, at face value you can say that if you play around the IL-4 and interferon gamma, then you can modulate the system as required. Of course, it is not so easy as it is said. Okay. So, Th1 and Th2 balance has to be achieved. As I said, there is an cytokine profile characteristic of Th1 cell and characteristic of Th2 cells. Just to give an example how the cytokine profile will change the humoral immune response itself, that depending upon the cytokine dominating in that atmosphere, the interferon gamma can shift the response, even the antibody response, to IgG3, and IL-4 will shift the response to IgE type of antibodies. You may have some idea about the IgE type of antibodies. That is the most recently evolved antibody among all 5 isotypes as such, and currently it is linked, at least at present, it seems, to immunopathological condition, that is hypersensitive or allergic condition. On other hand, the balanced cytokine profile that is observed at the initial phases of immune activation is an IgM. Okay. IgM also has some advantages, considered as the most ancient molecule in evolution of the immune system. It has still some advantages, but some disadvantages as well. Very bulky molecule. Okay, so with this, let us consider the infection. Now, as a microbiologist, I will say that an infectious agent enters the system and it causes infection. Host has nothing to do with that. That was what believed earlier at the first golden era of microbiology. They came up with many discoveries that pointed that microorganisms solely are responsible for infection. Now what is being believed, hypothesized, is infection can occur only in a susceptible host. Okay. So, susceptibility is being function of effectiveness of the immune response. So, whenever there is infection, now we can say that host is lacking in some immune response as such. It is immunocompromised, and that is strongly supported in case of cancer. Cancer cannot arise in a totally healthy immune system. It will arise only when there is immunocompromise. So, damage to the host may be caused directly by microbial factors, or it can be through host factors. That again is very true for viral infection. You know that most of the symptoms in viral infection are because of the host immune responses, not because of the virus replication, not because of any viral product as such. Again, a classical example is that of the herpes infection. The very painful rash, that is because of the host immune response and very severe inflammatory response. So, to put in a very vague manner, I can say at this stage that if you suppress the inflammatory response to infection, like the example I gave, then you can be relieved of the symptom. And I know few cases at clinical level where the patient was treated with steroids, which are known to have very good, very effective immunosuppressive activity. So, that controlled the inflammation and that relieved the host, that patient, of all the infection and finally cured the infection as well. That of course, these are some standalone cases as such. Some microbial disease cause damage to host because overly vigorous immune response. A strategy used by some of the microorganisms as well as some of the tumors, the cancers liberate the inflammatory molecules. So, there will be inflammation and that brings more and more nutrients to the cancer mass. Okay. So, the pathogen or the parasite, let us say, either microorganism or the tumor cells, they are playing with the immune system. They are modulating in a natural way the host immune system. So, host damage is a function of immune response, is represented by an parabolic damage response curve. So, immunocompromised against overly exuberant immune response, what you need is an disease threshold, that is the normal. That is where you come with the immunomodulatory treatment modalities. Okay, coming back to pathogenicity again. Initially, we speak about microbial virulence. There is invasion. There are different battery of factors. There can be adherence, colonization, spread. There are aggressins. There are different molecules, mainly having enzyme activity, like the most common pathogen Staphylococcus aureus produces battery of enzymes, okay, like coagulase, hyaluronidase, the kinases and so on and so forth, that help it in the spread. Some microorganisms will evade the host immune system, maybe simply hide from the immune system, maybe change their antigenic expression as well. Okay. The specific or non-specific defenses, the escape from this can be achieved. And then there are organisms that produce the toxins, and microbial toxins are considered the most potent in the world, particularly secreted by the Clostridium species. You can say, bit fortunately, they are totally anaerobic nature, so they cannot grow in healthy tissue and secrete the toxin. Now, if you consider another parasite, that is tumor, you will see that the evasive mechanisms and invasive mechanisms are almost same. The molecules can be different. The cancer cells, I don't think, produce any toxins as such. Okay. Okay, besides these determinants, the microorganisms develop resistance. What are the mechanisms of development of resistance? It can be very aggressive one, they will liberate enzymes. So, there will be inactivation of an antibiotic or modification of the antibiotic, and that has been observed. The first antibiotic that was put into the therapeutics, that is penicillin. Penicillin resistance have been now very common, observed in many pathogens, and one of the mechanism is producing the enzymes. So, the beta lactam ring of the penicillin molecule will be opened up, and so it becomes totally ineffective. Okay. There can be alteration in the target site of antibiotic. So, the binding capacity will be reduced. Okay. That is observed for most of the antibiotics that have the target as protein synthesis. So, the binding efficiencies can be changed. The metabolic pathways can be altered to circumvent the antibiotic effect. Okay. Well, this again is a very ingenious approach of pathogen as such. Or there will be reduced intracellular antibiotic accumulation that goes with drug transportation across the membrane, that can be reduced, and so the increased efflux or decreased concentration, decreased permeability, that will make the organism resistant. You will see that all these drug resistance mechanisms have been adopted by cancer cell depending upon the situation. Fortunately for microorganism, this can be predicted, this can be detected. For cancer, it has to be studied case by case. It is an very, you can say, an personalized picture where the cancer cell is developing resistance to the anti-cancer agents. Now, so as I said, it's a battle between the tumor and the host. So there are two aspects that you should know. What is the tumor to host relationship? Against that, what is host to tumor relationship? What we speak here is the host immune mechanisms that are available with the host to overcome or at least stop the spread of the cancer. Against this, the tumor have different mechanisms like evasion, as I said, for microbial pathogenicity. Same thing here, a cancer can arise at immunologically privileged site, very drastic, you can say. The example is that of an retinoblastoma. That region is considered as immunologically privileged one. The cells cannot enter that site, the cells of the immune system, or for that matter another is the central nervous system. There is no direct trafficking of the immune cells in this region. So, if any cancer develops there, then it grows very fast because there is no resistance from the host as such, and finally the immune capacity of the host against tumor mass. So, it grows to such an extent that even if the immune responses have been activated, they cannot tackle such large mass of the cancer, like it happens in case of retinoblastoma or the what is called as brain tumors, where usually whenever detected, it is considered as the last stage of the cancer. Okay. So, it can evade the host immune mechanisms. What the host should do in this case, it can be an intervention to aid the host immune system to activate the effector mechanism. Is this evasion can be overcome. For example, if it is at immunologically privileged site, then surgical intervention can make it exposed to the host immune system, and that will result into an immune activation, a specific immune activation. So, one of the present cancer therapies is an surgical removal. You remove the surgical mass, so you reduce the cancer mass. So, the host immune system can tackle better with the now less cancer mass as such, but it also opens up some of the barriers where the immune cells can reach there and further activation can take place. Okay, antigenic modulation, as I said, many of the cancers at the initial stages are going to express the antigen, and what we call this as tumor specific antigen. Tumor specific are different from tumor associated antigen in the sense that these will be specific only for the tumor and will not be present under normal circumstances, and these can be used for diagnosis and maybe for targeting the cancer tissue as well. But in the initial stages the antigenic expression will be low, okay, and that makes it difficult to detect the cancer, make the diagnosis, and on the host part to activate the immune mechanisms. There are some cancers that tolerate the induction, okay, that induces the immune tolerance. There is an hypothetical third type of T cell that is simply called as T suppressor cell, which is now partly recognized as T reg cell. That is, that brings about the regulation. When immune system is initiated, it has to come to an end, and that is supposed to be the function of the TS cell. So instead of activation, the cancer will induce an tolerance. That is, in other words, it will suppress the immune system, okay, and that can be an antigenic modulation. As I said, there can be shedding off of the antigens, or well, that is observed in more aggressive cancers, internalization. The cancer antigen, tumor specific antigen is expressed on the cell surface. There will be an antibody response, and vigorous antibody response. The antigen antibody complexes are formed, and these will be internalized and will be digested. Antibodies are proteins, so that's a good nourishment for the cancer. Well, I already mentioned immunosuppression, and certain molecules have been identified here, or this is more aggressive one, it induces apoptosis in the effector cell. Okay. Okay, what we have with the host is the effector mechanisms. So we already considered these. The humoral ones, this can be non-specific, like inflammatory molecules, the complement, the cytotoxic molecules, the ADCC, antibody dependent cell cytotoxicity, activation of NK cells, and so on. All these can be non-specific. All these can be the activation of the innate immune responses, the second line of defense. Cell mediated, there will be direct cell lysis by activated T cells, ADCC I mentioned. The cytokine mediated activation of the cells, we will come to that bit afterwards. The activated macrophages, the NK cells, and so on. So there is battery of the defense mechanism with the host, only that there was a tumor that itself indicate that this was not sufficient to initiate the cancer and then let it grow in large masses. Okay, so now we come to cancer chemotherapy. There are immune adjuvant, and lot of work was done initially which are non-specific, some are microbial. The Mycobacterium bovis, you know, that is a component of the old BCG, okay, that is known to activate the cell mediated immune responses, as we have seen in our basics of immunology, that the cell mediated responses are specific as well as it activates non-specific cells like macrophages, the platelets, mobilizes the bone marrow, and so on and so forth. Okay. So that can be achieved with Mycobacterium bovis through BCG. Levamisole initially and anti-Hillman drug was found to have an immune stimulating activity. So that was used earlier. There are mitogens that have been used. Some of these were chemically defined, and now it is known that plant extracts may have mitogenic activity as well. Some are used very classically in immunology experiments like PHA, phytohemagglutinin, and so on. Okay. So that will have mitogenic effect. So whenever there is immune activation, we need to expand the clones, and for that such mitogenic molecules will be effective. Cytokine therapies have been used. There are commercial preparations available for last 2 decades. I haven't put any slide here, but there are such commercial preparations available which will include variety of cytokines. One such is interferon alpha, interferon gamma more popularly. There is TNF alpha, TNF beta, or there can be IL-2, okay, or in some cases GM-CSF. All these are available. These cytokines will activate the innate mechanisms and very effectively can bring about the tumor suppression. Only that they are used for local application. Okay. Their systemic use of cytokine is associated with serious, sometimes life-threatening consequences. There is a disease condition associated with unregulated release of cytokine, and that is called as an toxic shock syndrome or scalding skin syndrome as such. So cytokines are not given systemically, and it has to be monitored very, very carefully. Otherwise, in a patient who is already immunocompromised, it can result into an life-threatening consequence. The in vitro activation that has been practiced in some areas of world, particularly in UK, that you take out the tumor and you take out as well the dendritic cells, one type of macrophages from the patient itself. Treat the tumor maybe with chemotherapeutic agent or irradiate it. So it will not grow, but it will remain alive, and well, it will still keep its antigenic expression as it is. So these patients' own dendritic cells will be interacted with the excised tumor, and then by using the immunomodulatory agent, okay, let us say simply by using PHA, phytohemagglutinin, a known T cell mitogen, these macrophages will be activated, and then these will be passed back to the patient. And it has been observed that this type of treatment modality has worked very effectively and can bring about regression of tumor. The advantage of this is the tumor recurrence is not observed in such cases, because the characteristic feature of the immune cell is memory. They remember, and so very effectively they can control this. Sir. Okay. So the tumor infiltrating lymphocyte, that is based on the observation that the tumor is infiltrated by the lymphocyte. Why they should do that? Okay. But then there must be some specific mechanism involved. That is still being worked out, but they have already put in some practice. The lymphokine activated killer cells or tumor infiltrating lymphocytes, they are used for such type of in vitro activation and building up an very effective immunotherapy. Initially monoclonal antibodies has proved to be a great promise, But as I said, tumors go on changing their antigenic expression. They modulate their antigenic expression. So what you can identify the antigen, raise the antibodies against that, maybe couple that with the toxins or other cytotoxic agents, okay. And if the tumor changes its expression, then all this goes west. So what is to be done? Immunomodulation, taking clues from Indian traditional system of medicine. Okay. As sir mentioned, there is a class of Ayurvedic preparation, that I think the word comes from the single chemical entity, SCE. They work like a single chemical entity. I don't know whether this comparison is right or wrong, okay. But these are rasayanas that have shown to have an immunomodulatory activity, and that was revealed by the studies carried out here, and most studied was the Withania somnifera. Okay. Well, it is now recognized that immunomodulatory therapy for a variety of disease condition. When you say disease condition, as we said earlier, it itself implies that there is an immunocompromisation to some extent or other. So when host defense mechanisms have to be activated under conditions of impaired immune responsiveness, or when a selective immunosuppression has to be induced, like inflammatory disease, autoimmune disorders, cancers, or bone marrow transplantation, such immunotherapy will be more promising. Well, I know there is a lot of information here, and you won't have a chance to go through this all right now, but the presentation will be here, so you can take your copy. But what I want to mention here is Withania somnifera. The potential of this was recognized long back by modern age scientists in 1958, and he referred this as an adaptogen. Different parts have been tried, and the leaves are shown to have some withanolides that have direct interaction with the tumor and may have to some extent anti-cancer activity, but more studies have been found it as an adaptogen, and most of the time it is the root. It has been used for a variety of conditions in folk medicine, in traditional medicinal practices. There are a lot of papers. There is a long list of papers. Many of these are from this center itself, and I haven't gone beyond 1999. Okay. After that, I know that a lot of significant impressive work has been carried out. So in conclusion, I should say that immunomodulation will be a very effective means and will be the treatment of the future for any disease condition, let it be arising from the body itself, like the autoimmune condition, it can be cancer, or something coming from outside, that is an infectious condition. That's all I have to say at present. Thank you.
These rasayanas or these observations what we have on immunological effects of some of the plants, can they be useful to modulate the transporters? And that's what we are going to do. Dr. Sisodia may have already shared those ideas with you. So this is a new area which is emerging, and like you are able to modulate immune response, it is possible to modulate transporters also. And some studies on materials like curcumin already have shown some promise that, and if you are able to modulate drug transporters, impacts, as you know, on the entire AGMET. And even if you are able to, for example, modulate the modulators to improve the pharmacokinetics or bioavailability of the drug, you can imagine what kind of impact it can have on the therapy. Because one of the major problems is cytotoxicity. So you are inducing cytotoxic drugs. If you are able to reduce, for example, in tuberculosis, if you are able to reduce the dose of tuberculosis by improving bioavailability, by modulating transporters, it's a new area which is of interest, and that was the purpose to get that on board on the light of the background of increasing problem of drug resistance or multi-drug resistance that we are facing, which is also now applicable to cancer through resistance coming to cancer drugs. Now, if you have any questions, we'll be taking some of them now. Yeah. Okay. Thank you, Dr. Diwanay.